Hi, we're back here again with my friend Rob Stadler, who is no stranger to this channel. What he does is he complements my background, which is more on the organic chemistry side and the origin of life, and Rob understands a lot more about the biological side, the evolutionary side, what happens once you get life. Rob has been trained both at Harvard and MIT in a dual program, has a deep medical background as well as biology and chemistry. And he, for over 30 years, has been working in the medical device area, and his work is implanted in many people these days. So he's had a really large contribution, predominantly from the industry. And with that, I want to turn it over to Rob to talk about long-term experiments show that evolution is highly constrained. And so if you could start by telling us, Rob, what that title means, and then start to walk us through it.
Yeah, thanks, Jim, and thanks everyone for joining. I've been eager to do this for months now, so really excited to be here. We're going to see today very clearly, as I think as clearly as science can present, that the process of evolution, specifically random mutations and natural selection, which are given credit for all of the diversity of modern life, we're going to see how constrained that process actually is. We're going to look at the highest quality and most recent evidence. When I say high quality, I mean this high confidence evidence that comes from prospective experiments conducted recently. And we're going to see that paints a picture of how constrained the evolution process actually is. That's what we're going to go through today. And I wanted to start off by giving a little insight into the researchers that are doing this kind of work, these difficult experiments on evolution, and what they're doing, what they're saying, what they're thinking. Because these scientists that are doing this work, they actually recognize, as you'll see in the text I'm about to show, these quotes, they recognize that it's extremely difficult to demonstrate any kind of innovation coming from random mutations and natural selection. They believe in their hearts that that is the process responsible for all of life. And yet, as you'll see in these quotes, they're scratching their heads wondering why we can't demonstrate it experimentally. So here's a paper where there's a quote, "The tree of life is punctuated with numerous biological innovations, but explaining the origin of novelty has been problematic." So you have to scratch your head here because it's been more than 160 years since Charles Darwin wrote his book, The Origin of Species, and he promised us that future evidence would come forth to make it clear why variation among species and natural selection are able to explain the origin of biological innovation. And here we are 160 years later, and the researchers are still scratching their heads about this. Thus, understanding the evolution of complex innovations remains a formidable challenge. 160 years later, folks, still a challenge. How new traits originate from evolution is a fundamental question of evolutionary biology. Why is it still a fundamental question? Can anybody think of a theory from 160 years ago that made a claim and we're still struggling with that as a fundamental question, still struggling to explain it? More precise statements about how tinkering happens, the genetic mechanisms that generate a novel function, and the ecological conditions that promote its origin and spread remain elusive. 160 years of trying to find it, and we still say it remains elusive. This should cause a lot of concern. And today we're going to go through, some of the evidence that they're talking about here, and we'll see that the work they're doing is painting a very nice picture that random mutations and natural selection just can't innovate. So to take a step back and look at the big picture again, what we're doing here is trying to find out, according to science, which of these two hypotheses is better supported. Did all of life come from a tree of life, a common ancestor producing all of the great diversity of life that we see today in the modern world? Or alternatively, is it more of a forest of life where we have separate trees producing related to separate kinds of life? Each one is able to diversify within its own tree, such as the dog, the wolf producing all the different breeds of dogs we have today. It's a much more complex, diverse tree than we show here in this cartoon, but you get the idea. So which of these is preferred by the science? If you're a good scientist, you open your mind to follow the evidence where it leads. You accumulate the evidence, you stack it up, and see which one is supported. And when we accumulate the evidence, there's two particular things we need to look for in evidence here. And one is two criteria of the evidence. One is we want evidence that's able to distinguish between these two. If the evidence is equivocal in supporting either one, that's not useful. We don't care about that kind of evidence. We want evidence that separates them. And the second thing is we want to prioritize the evidence that gives us the highest confidence. We don't want evidence that gives us low confidence. And for those who that term is new to, this is a topic of my first book, where all of science, you can determine the level of confidence that evidence provides by considering these 6 criteria. Does the evidence meet these criteria? If so, then it provides more confidence. We're looking for evidence that can be repeatably generated. We're looking for evidence that is more directly observable. The more directly you can observe it, the more you can have confidence in it. We want evidence that's obtained through prospective study. So experiments designed in advance, and you prepare, you can block out any kind of confounding factors. You have a control group, and you can actually determine from this causality, like this caused that. And we want results where the investigators are careful to get their bias out of the picture, and we want minimal assumptions made, especially assumptions that are hidden and are not disclosed. And we want to see that the claims that are produced from the evidence are reasonable claims that address the way the experiment was conducted and the evidence that was collected, and not extrapolate far off into some other area. That's what gives us confidence. And as you may have seen from my previous presentations, the type of evidence that's often used to support evolution, the single tree of life, this is evidence that's in our school textbooks and presented in all kinds of media: the fossil record, the distribution of different life forms across the world, vestigial organs, and then comparison of life forms, like comparing their DNA or comparing their morphology, the way their limbs are structured or anything like that, and showing like a phylogenetic tree, a high correlation between some forms of life and a correlation structure. All of that kind of evidence, it fails to meet each one of these 6 criteria. So it belongs down here in this very low confidence side of the scale. What we want is evidence way over here that provides high confidence meeting all of these criteria. And today we're going to go through a series of experiments conducted recently, testing what evolution can accomplish. And those are all meeting these criteria and giving us higher confidence. That's what we're going to do today. So again, as we look in comparing these 2 hypotheses, each of them provides a prediction. This one, the tree of life, large-scale evolution, gives us the prediction that random mutations in natural selection are able to produce innovative new functions. Modern life has millions of innovations, and they had to come from random mutations in natural selection. That's the ultimate source of evolution. We're over here, we're going to predict that random mutations in natural selection can cause some minor adjustments, some tweaks, but they really can't produce innovative new functions that would, get you from one tree to another tree or break out of the tree of the kind of life that you're in.
Yeah, and Rob, let me mention that in case people haven't seen it, you and I have done a couple of other videos where you have contrasted these 2, all life coming from a single cell, which was the progenitor of all life, to this forest of life, where it's this one kind was made and then another kind and another kind. And so if people have missed that, we have several predecessors to this particular video that go into a lot more detail on that.
Yeah, thanks for that reminder, Jim. Appreciate that. And so the data that we're looking for today, in order to try to separate these and find out which one science really supports, We want high confidence evidence, not low confidence. And we want to see innovations developing from random mutations and natural selection. That'll help us to separate these two. And when I say innovations, that's where things become a little subjective. What do I mean by biological innovations? And previously we talked about this question of, could evolution ever receive a US patent? This is our society's way of kind of judging if something is innovative or not. Is it worthy of a patent? And in the US, getting a patent means the idea has to be novel and not obvious. Novel meaning that it hasn't been disclosed before, you can't find it in any publication or a previous patent. It's new. And not obvious is a bit more subjective, and it means basically it's not just a minor tweak. As we mentioned in a previous video, it's not like just adding another wheel onto a wheelbarrow. It's something more innovative. Life is based upon millions of innovations that could easily achieve this kind of status of a patent. We're just asking, can random mutations in natural selection produce a patentable idea? And for those who think this is sort of too subjective, too hard to follow, we're just going to look at the types of changes that random mutations in natural selections have produced in experimental studies. And when you compare those against the kind of innovations we're going to show you that life requires, you're going to see quite a discrepancy. And that you can use that if you don't want to think about innovation in this way. So as Jim mentioned, in our previous video, we had this very nice looking thumbnail up here. It's kind of the best I've ever looked, I think. But in this study, in this presentation, we went through this review paper published just this year in Nature, where they reviewed long-term evolution studies, the type of data we're talking about here. And in particular, they covered these 6 studies that we reviewed. And in reviewing these studies to see what random mutations in natural selection could have produced or could innovate in these 6 studies, we found again and again that there was really nothing innovative here, nothing that random mutations in natural selection innovated. So I'd invite you, if you haven't seen that, to go back and review that video. But today we're going forward because there are many more studies outside of this review article that we covered that are very important to this topic. So these are new studies beyond what was presented there. And the first study happens to be my favorite. This is a beautiful study where the investigators purposely tested evolution by purposely breaking a gene and seeing if evolution can fix the problem. It's a beautiful study. So E. coli, the bacteria E. coli, are able to produce the amino acid tryptophan. And you should be jealous about that because us humans, we can't produce our own tryptophan. We have to eat it in order to get it, and E. coli can make their own. And it's a complicated process, but one of the enzymes used to produce tryptophan is shown here, tryptophan synthase. And that's what the tryptophan molecule looks like. So these investigators said, hey, if I purposely damage this gene, if evolution came up with this gene, certainly if I put a little nick in it, if I cause a little damage to the gene, then evolution should be able to repair that problem, right?
Can I just mention something? That tryptophan synthase, you don't mean that it is a gene. You mean that the gene that prescribes the building of the tryptophan synthase, correct?
Exactly, yeah. Thanks for clarifying that. So this is a protein. It's actually four proteins, 2 of the same kind, that join together to make this enzyme on a protein, but it's coded for in 2 genes. Good point. So they modify the genes, and therefore the produced protein has been modified and less functional, and they're going to test to see if evolution can fix the problem. So in their first experiment, what they did was they changed just one letter, one nucleotide of the DNA, and that was in one of the genes. And now the produced enzyme functions poorly. It can still produce tryptophan, but not nearly as well as it should. And then they put that mutated E. coli into a solution where there was a limited amount of tryptophan. So there's a selective pressure to develop the ability to produce your own tryptophan, and then you could take over the population and be the champion. And so they did this, and what they found was sure enough, after about 100 million E. coli were grown in this solution, a random mutation, supposedly a random mutation, fixed exactly the problem. It changed that letter of DNA back to the correct letter. The enzyme was now fully functional, and those bacteria took over the population because they could make their own tryptophan. Wonderful. So we see a success story for evolution, for random mutations and natural selection being able to fix this problem. So they tried another one. In the second experiment, they changed a different letter or nucleotide of the DNA code. And now they ended up with a completely dysfunctional gene, a completely dysfunctional enzyme. It wasn't able to make tryptophan. And they put it in those same experimental conditions with limited available tryptophan. And once again, after about 100 million E. coli were produced in the solution, the problem was fixed by random mutations. And the E. coli with the repaired gene were able to make their own tryptophan. They took over the population. So once again, we see repeatably that evolution can fix that kind of a problem. So then it gets more interesting when they did their third experiment, where they had both of the DNA changes. So two nucleotides of the DNA were modified. And again, we have an enzyme that is not functional. But in order to get the function back, you got to fix two problems. And in this situation, they used the same kind of media with limited tryptophan. And they actually grew 9,300 generations of the E. coli in this media. And we're talking about like 1 trillion total organisms produced here. This is more than there are human beings, of course, and I would argue more than there have ever been, this many human beings in all of time. So it's a whole lot of organisms, a whole lot of opportunity for evolution to try to get a job done. And what they found was that, unfortunately, the function of the tryptophan synthase was never restored. And not only that, but the genes that are used to produce the tryptophan synthase were either deleted or simply suppressed. And the reason why that occurred is that evolution here is taking a shortcut. It's basically saying that producing faulty genes, producing faulty enzymes is using up tryptophan. And the tryptophan is a precious limited resource. So we're better off fitness-wise to just stop producing these broken enzymes. And we're more fit and just give up on that. And that's what evolution did. It took a shortcut rather than fixing 2-point mutations. So we certainly don't see any innovation here, certainly not worthy of a patent. And that's what this experiment showed.
If you do not believe in the physical resurrection of Jesus Christ, send me an email, tour@drjamestour.org, and we will get together and I will share with you about why I embrace the resurrection of Jesus. Can I just say one thing here about this so that our audience understands? What happens is he has this long string of DNA in the genome, and that codes, the DNA goes through RNA and then codes for the building of these enzymes, which is the little colored thing in the top left of what he's showing now. And it's those enzymes that build the body. So what he did is he chose an enzyme that makes the amino acid tryptophan. Then what happens is, what was shown is that you can modify one letter in a very long string, one letter, and yes, the organism can go back and correct that and fix it. But if you change just 2 letters, what's produced has no functionality, and all this evolutionary pressure through many generations, it can't be fixed. So even something as small as just 2 letter changes can no longer be fixed. And what the organism is now doing, it's saying, let's just totally stop using this segment, not even try to fix it anymore, because in the meantime, we keep making this enzyme that does nothing. So this is showing that if you want to have a gross change, a gross evolutionary change, evolution can't do it itself. All these changes that scientists like to talk about, well, this changed into this, that's a red herring. It's not happening. If you get more than 1 letter change, the thing totally shuts down, and no natural evolutionary process, even when the system is trying to be forced into this, it can't correct it. Have I interpreted that right, Rob?
Yeah, I might clarify that there are situations where you're going to see more than one letter change. Um, it becomes we'll go through some numbers, and it becomes
Okay.
extremely difficult, but not impossible, especially if changing one letter gives you some kind of advantage, and you can then change the next letter and get another advantage, and you have these sort of small stepping stones, then I think things can be accomplished that way. But that's a very constrained route to get something done.
Mm-hmm.
Not much will get done that way.
Okay.
So to put some numbers on this, there's actually about 2,000 nucleotides for these two genes that make up this enzyme. And we have 2 letters that are wrong. So it's actually 99.9% accurate genetically. And we said, okay, evolution, take this to the finish line, and it failed. So then how can you believe that this entire these 2 genes came out of nothing if they can't even get you to the finish line when you start off 99.9% complete? And it turns out to be worse than that, because the actual biosynthetic process to make tryptophan is actually 5 different enzymes. Starting with Chora's mate here, there's 1 enzyme, 2, 3, 4, 5 different enzymes with their intermediates in order to get to tryptophan. And we've only been talking about this last enzyme here. And if you look at all 5 of these, there's about 6,800 letters of code. And we've shown, this study has shown, that if just 2 of those letters out of 6,800 are wrong, then the whole process is scrapped. So to give you a helpful analogy here, imagine running a marathon, right? 26.2 miles, you're going to run the marathon. And we're actually going to give you a head start here. Our runner is called Evolution. And we give Evolution a head start so that they only have actually about 40 feet left to run. They get to start 40 feet away from the finish line, because with only 2 errors out of 6,800, that's like 99.97% finished. And they start this race 40 feet from the finish line, and they trip and fall and give up. And yet our society gives evolution the credit for producing this entire complex structure. In other words, finishing the whole marathon when it can't actually finish from 40 feet away from the finish line. And that's not the only study. Here's another study. This is with yeast, so it's eukaryotic instead of prokaryotes like E. coli. But they went through 10,000 generations of yeast, and they put these yeast under 3 different conditions. We don't need to go into the details, but there's 3 different situations they were placed in. And again, this is all this high confidence evidence. So they did this repeatably with many different individual populations evolving separately, and many generations grown each day. And here's what they found. These graphs show the fitness over time. You see up to 10,000 generations of the yeast, and things are getting more fit over time in each of the 3 different treatment groups. And they explain it as with other studies, populations predictably increase fitness rapidly in the first few generations, and then there's a slower adaptation over time as time progresses. So what this is showing us basically is this rapid increase in fitness initially is not an evolution process per se. It's not random mutations and natural selections. It's actually redirected use of existing functionality. So it's like the cell has a really complex control system like this, the control system of a nuclear reactor. And it gets placed into a new environment, and suddenly some light goes off, some warning, beep, 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 something's out of range here. And the control system and the controllers are very intelligent. They're able to flip a few switches to adapt for that new situation, that new environment. And they're able to get function back in working order pretty quickly, although not perfectly. And then they make, over time, a few little adjustments, a few tweaks, a few turns of a dial, and things get better and more optimized over time. And that's kind of what you're seeing in the cell here. Quick adaptation and then some slow optimization process. But what's really interesting in this study perhaps those that are looking closely, you see these individuals, not individual cells, but individual groupings of cells separated out that have the asterisks here. There's 2 here, 3 here, 1 here, that are really the champions. These are the best yeast in the contest. And so what's going on there? They're really blowing this out of the water. So there must be some kind of major evolutionary innovation going on here. That's what we're really focused on today. And so what we find is the asterisks in the paper. You could read this. Here's the reference. But the asterisks actually mark populations that had a single point mutation occur that was not actually innovating, but it was actually fixing a gene that was broken in the founding population. This is a well-known point mutation in yeast W303, which is what they started with. And this point mutation makes it unable for the yeast to produce its own adenine, which is another molecule that's needed for life. So this dominant fitness benefit we're seeing here is actually fixing a single bad nucleotide. It's not a type of innovation. And we're going to dig into this because it's fascinating to see this process. Remember, we just went through this whole thing with tryptophan with the 5 enzymes, how complicated it was, and how 2 point mutations couldn't be fixed by evolution. Well, the process of building adenine is fabulous, fantastic. So I love this drawing. Here's the reference for it. But this drawing is showing us a portion of the biosynthetic pathway to produce adenine. And in here, we start with ribose 5-phosphate at the top. And you need this fancy protein enzyme in order to catalyze the reaction to get to the next intermediate. And then it's catalyzed by this enzyme to get to here, and this enzyme to here, and this one to here, and this one to here, and this one to here. And there's on and on and on. There's 11 enzymes, each of fantastic complexity. You can see here 11 catalyzed reactions. And we're still not at adenine yet, but we're very close. So there's a few more steps.
This is wild. This is wild. I was once looking over the synthesis of the simple molecule glucose, and it was something like this. It was like, 11 or 14 enzymes, and there were several coenzyme steps in there, and I presume some of these have to have added coenzymes as well. This is not going to happen by some random process. I mean, each one of those colored entities had to have been encoded in a gene, in DNA, and then transferred that to an RNA, which transferred that to the building of each one of these. And these steps are just monumentally hard. And to come up with an enzyme that's going to carry out that step is really hard to fathom. And it's things like this that evolutionists will just look at it as if it's no problem. These things will just modify and form this way, and you'll end up getting adenine at the end of the day. Rob's about to take you through one very small step along this and show you how incompetent an evolutionary model is to be able to accomplish this. Rob, if I got that right?
Yeah, that's a good word for it, incompetent. I like that. So what we're going to do is focus in on this section up here of this 11-step process. And what happened is the yeast that they started with, this W303, it actually had one nucleotide that was wrong, and it's in this enzyme right up here. Again, the DNA coding for this is where the problem was, and that led to a protein being produced that's wrong. In fact, it was a rather devastating mistake because it converted what should be a glutamine amino acid in this protein to a stop codon. So in other words, in the production of this complex enzyme, there's a command given to stop producing right here. For those who like details, this was the 65th amino acid in the series out of 571 needed to produce this one enzyme. And so those of you that know how assembly line processing works, we have a broken enzyme here, and the entire assembly line is broken. Not only that, but what we end up doing is building up this intermediate because we're stuck here. We can't go further from this intermediate. And it turns out that this intermediate is actually a little bit toxic. It's a mild toxin. It's kind of interesting historically because this turns the yeast red. And so people notice that and have studied this for a while. Why is the yeast turning red? Well, it's because this toxin is building up. And so this yeast, you have to feed them adenine or else they can't survive. They can't make their own adenine. So what's interesting about that is building up this toxin. What's interesting about that is now you don't want this toxin. And so the yeast is going to try to fix the problem. Well, you could try to fix the problem by randomly fixing this issue over here. And that's not very likely to happen, right? What's much more likely is that you'll have some kind of breakage upstream in the assembly line. Something else is going to break, like this part right here is going to break, and that will stop the assembly of the toxin. So there's actually a benefit to the yeast in order to break the assembly line upstream. There's a fitness benefit to that. And there's only one way to fix the problem, and there's lots of ways to break upstream. So out of 205 replicate populations, 6 of them, those are the ones with the asterisks, they were lucky enough to fix the problem here, but the rest of them all broke something upstream. And once an upstream pathway is broken, evolution really can't fix it. And here's the problem. It's because if evolution fixes this, then what do? You end up producing the toxin. So you're worse off. That's not going to help. And if evolution only fixes this guy over here, that doesn't help whatsoever because it's already broken upstream. So you really have to fix both of these rather simultaneously in order to get the big benefit. And as we've seen, evolution really isn't capable of that kind of 2 simultaneous fixes. So here's a quote from the paper that we've been reviewing. They call this, when you have 2 problems like this, they call that the lower fitness genotype. And they say, we do not observe any population that moves from the lower fitness genotype, having 2 errors, to the higher fitness genotype, meaning 0 errors, even after 10,000 generations of evolution. Okay? So once again, we have a problem where evolution is given the credit for producing this incredibly complex series of 11 enzymes. And yet we find that 2 simple errors make it basically an unrecoverable process. So how can you believe evolution would have produced all of this?
Because of you, millions of people have heard the gospel. If you confess with your mouth Jesus is Lord and believe in your heart that he's risen from the dead, and you will be saved. That's right. That's the requirement. We talk about science concepts which draw people in.
Take these nanomachines and have them drill into cells.
This would be a great way to kill cancer, right? We also talk about Jesus Christ, who's the best in everything.
My faith in Jesus Christ means more to me than anything.
If you could continue to give or give for the first time, we would certainly appreciate it. You can go to jesusandScience.org/donate. All U.S. donations are tax deductible. Thank you so much. Yeah, this is really striking. I mean, we do give an enormous amount of credit for evolution. You just say evolutionarily, this happened, and everybody is programmed to back off you. Oh, evolution solved this. But this shows you when you have these 2 little errors, there's nothing that evolution can do to get us past this. And now, how could you have come up with this entire sequence to make this adenine? So this is it's really striking about how little evolution is able to do.
Yeah, and to give your word on it again, it shows incompetence, really. I mean, we're seeing sort of random bumbling, which we know can maybe sometimes benefit somebody, but overall, no innovation and basic incompetence, what we're seeing. And I throw, this is a little sidebar here for adenine production. I'm going to throw this in because there's another aspect to this. Adenine is also the fundamental building block for how life harnesses and uses energy, because adenine is used to produce ADP, adenosine diphosphate, And these are the so-called empty batteries of life. You have to produce these empty batteries, and then you charge the batteries up to make them ATP instead of ADP. And the ATP, the charged batteries, are used in life to do all kinds of processes like moving your muscles and replicating and fixing things and everything that life does. So ATP is the energy behind life. But here's where the trick is. To produce one molecule of ADP, you got to go through this process. And this process, as you can see, uses a lot of ATP. In fact, you need about 7 molecules of ATP in order to produce one ADP, the empty battery. And so what we're seeing is a chicken and egg phenomenon, that this process couldn't have started without ATP, but you don't have ATP until you have this process. Now, we humans are kind of able to figure out chicken and egg problems like this. I'll give you an example, like our cell phone situation. Cell phone towers, which bring the cell phone signal all over the place, they have no value unless you have a cell phone, right? And the cell phone by itself has no value unless you have the cell phone towers. So random processes wouldn't have thought through with the foresight to produce the system we have today. But humans with foresight can say, yes, I'm going to build the towers and I'm going to build the cell phones, and the whole thing works as an integrated system. And that's what we're seeing here, something that's integrated system to overcome this chicken and egg problem. And I think there was foresight behind it. Okay, so we're going to switch gears into a third study here. And in this study I'm pulling out because someone in the comments of our previous video said, hey, you should review this one because this one shows innovation coming from evolution. So here it is. The title of the paper is Random Sequences Rapidly Evolve into De Novo Promoters. In other words, functional sequences. They wanted to see if random sequences of DNA could produce an innovative function. And as we'll see, the answer to that question is it depends on the complexity of the function. If the function is something really simple, like having a doorstop, if you look around your room right now and look at random objects in the room, how many of those could be a functional doorstop? A fair amount of them, right? But how many random items in your room right now can serve as a useful calculator? Maybe 1 or 2 or 3, not much. That's what we're going to see here. So what they did is, starting with E. coli again, and here we're looking at E. coli's ability to metabolize lactose, which is the sugar in milk. And in order to do that, E. coli has this thing called an operon, which is several genes in a row. And these genes are here to help metabolize lactose when called upon. E. coli would rather not eat lactose unless it's really necessary because it's a little harder to digest, let's say. So upstream of these genes is what's called a promoter, and this is where the transcription process gets kicked off by a sequence of a promoter. And the DNA sequence for that promoter looks like this. It's 6 particular letters of DNA, and then there's a spacer with about 17 nucleotides or bases, and then there's another 6 particular letters. So this is what the promoter is, but the promoter actually, it also allows many variations. It doesn't have to be exactly this. In fact, in some cases, you don't even need these last 6 letters. You could just have the first 6. So it's variable. And what they did is they stripped out 103 nucleotides out of the bacteria, and then they inserted random DNA sequences of 103, and they just plopped them in there. There was 40 of them that they tried out. Plop them in there, and then see what happens when you grow the E. coli that are mutated like that. You grow them on a combination of lactose and glycerol. The E. coli would much rather eat the glycerol. They eat all that up first, and then they have to say, okay, let's try to eat the lactose. And we see if the bacteria are able to do that. And the surprising, very interesting finding from this study is that after less than 100 generations of E. coli grown, they had 95% of those 40 random sequences were actually successful to provide the ability to metabolize lactose. That's kind of cool. And you might say, well, that's quite an innovation. But when you look into actually what happened genetically, we get this pie chart of the 40 different random sequences of what explains what happened here. 10% of the time, so that's 4 of the 40 sequences, the random sequence itself was already able to meet the required promoter specification. So 103 random letters already met good enough specification in order to produce the lactose enzymes, and it was successful. So that tells you, like I said, if you look around your room, I'll bet 10% of the stuff in your room could be a useful doorstop. So it's not a very specific requirement here. So the next big chunk, 57%, turns out that only one point mutation was needed in these cases in order to have the ability to metabolize lactose. So one letter was off in the random sequence. One letter had to be changed in order to meet the specification to metabolize lactose. You can see this is already the majority of the cases. So either 0 mutation or 1 mutation gets the job done. So the guys that are left here, these must be more complicated. These must be like 2 mutations, right? Well, we've already learned that 2 mutations is pretty challenging. So this 5% here, turned out they never developed the ability to metabolize lactose. That's the 5% that couldn't get the job done at all. Then we have 12% of the sequences where something else happened. Each operon like this has a termination sequence that separates it from the neighboring genes, the neighboring operon. So when this gene gets transcribed into RNA, you need to have something that says stop here, or else it'll just keep, using the next genes in the series. So there's these termination sequences here and upstream here. And what happened in this 12% is the E. coli deleted the termination sequence up here. So use of the previous genes meant it was also using the lactose genes. Okay, so to give you an analogy there, it's kind of like having 2 light switches on your wall, and one light switch controls the left lights in the room, and what other light switch controls some other lights in the room. But what the E. coli did here was merge those 2 light switches into one switch. So one switch controls all the lights. And hopefully you can recognize that as a loss of information. It's a loss of control. It's a kind of a devolution instead of an innovation. And that happened 12% of the time. And then in the final category here, we have something else happened, which is not completely described in the manuscript. I don't think they fully understood it, but somehow a promoter in a different area of the genome was moved and dropped in here to do the promoting of the lactose genes. Okay, so the one thing you don't see here is two or more mutations happening in order to get the sequence to work out. So it's very similar to what we've already seen. 2 or more simultaneous mutations, especially if there's no fitness benefit from the first one, is really, really hard for evolution to do. So if you have a function that's really nonspecific, like a doorstop, such that 10% of random sequences can do that job, then evolution can do that job, But if you need something more complicated, like you need a calculator, something complicated, it looks like 2 or more mutations are not going to happen. What evolution is going to do is find a shortcut. It's going to knock out something or kill something in order to sort of halfway get the job done and have a fitness benefit, even though it's not innovation. So just to step back to the 30,000 foot view. We've gone through 3 studies now that are painting us a very clear repeated picture, and it's the same picture, that achieving 2 mutations, if you have minimal fitness gain from the first one, is really, really difficult for evolution. For tryptophan synthase, we couldn't achieve 2 mutations, even with 9,300 generations. It actually just took a shortcut and either deleted the gene or just repressed the expression of it. With the lactose situation, again, we couldn't get 2 mutations. It was just too hard. And then with the adenine, the very complicated process to produce adenine, 2 mutations were really enough to shut the whole process down. And even with 10,000 generations of yeast, they couldn't find a way out of it. So I hope you're seeing a consistent picture here. In fact, it's so consistent that there was a paper written about this. This is in Genetics 2008. And it's a paper called Waiting for Two Mutations. And in this paper, this actually is not the high confidence experimental evolution that we've been talking about. This is actually more of a theoretical paper with a lot of math. But I throw it in here because it's speaking to exactly what we're finding from the high confidence experimental studies. What they tried to do here theoretically is say, what would it take for two mutations to occur without a benefit from the first mutation? And those two mutations would be turning off regulation according to one transcription factor and then turning on another one. So 2-point mutations. And what they concluded in their work here is that if this were to happen in human beings, our population, achieving two coordinated mutations for humans would take more than 100 million years of evolution. Okay, and so appreciate the fact that humans, no one thinks that humans have even been around for this long, ever. And so it means that this is not possible for humans in our population. That's pretty telling because humans compared to chimps, we've talked about this previously, we're talking about millions of DNA changes between humans and chimps. And we're saying two of those, if they need to be coordinated, are just not possible. For those who are interested in numbers, I'll go through this quick, but the entire human population today is about 8 billion people. That's 10 to the 9th power. That's our population. In order to have a mutation, a point mutation occurring in a particular location, the chance of that kind of error happening is roughly 1 out of 100 million. If you want a particular point mutation to occur, let's say the point mutation that causes cystic fibrosis, which is well known, randomly 1 out of 100 million humans will end up with that point mutation and cystic fibrosis. But if you want 2 point mutations, 2 particular mutations without a benefit from the first one, that would be 10 to the 16th power. Not just like 2 times this or half of this or something, but it's 10 to the 16th power, 1 out of 10 to the 16th, which is 1 out of 10 million times a billion. Okay? And there have never been that many humans ever in the history of all life. Never going to see that many humans. And that's why they came up with this conclusion. We see how impotent evolution really is here. Now, some of you might say, wait a minute, we know from Richard Lenski's experiment with his E. coli that we're able to metabolize citrate. Hey, we know from that 2 mutations were required, and it did happen. Well, there's a couple of exceptions here. First of all, bacteria are just prolific. You know that in Richard's study over 30 years, over 1 trillion organisms, individual organisms, have been produced in his lab during this process. And that's more than we've ever seen in human beings. There's just never been that many humans. And we also know that the first mutation in this 2 mutation series provided some fitness benefit. And we also know that the mutations he's talking about are mostly gene duplications, and those are prolific. Gene duplications are the most common kinds of mutation, happening at about 1,000 times more often than a point mutation. And so, yes, he achieved these 2 mutations, but there are some exceptions that made it possible.
Let me just mention that one of our other videos together is you took that Lenski work and you went into it in much more detail.
Yep. If you want to go deep, look into the previous videos and you'll see some depth to that. So this study I find really fun and really interesting because it involves rice developing the ability to produce in cold climates. So we all think of rice as being a tropical, this is China here, we all think of it as being a tropical product. But they found over a course of years, you could get rice to be productive even up here in the north in the cold climates. And I give my hats off to these investigators because they really worked hard on this for 10 years. It's a beautiful study and it shows that persistence pays off here. So they did a 10-year study and from year 1, they took seeds from the plants that produced the most seeds in cold climates. And they took those seeds to grow the next generation, which was also placed in cold climate. And they found that after only 3 seasons, there was the winning variety of rice that produced lots of seeds in cold climate. So the rice was able to adapt to this cold climate. And that kind of sounds like an evolutionary innovation. I mean, if you published a paper that said the rice evolved to acquire cold tolerance, I think you'd get that published, everybody would agree, and you could move on and claim victory. But these guys were much more detail-oriented and really wanted to drill down, find out what happened here. Is this really an evolutionary innovation? So they did extensive genetic testing comparing those that did adapt and the original rice that weren't adapted. And they, to their surprise, they found there were no genetic differences that were correlated to, this cold tolerance. And that was kind of frustrating. They had to dig deeper and deeper. What they did find, to bring a long story short, through extensive effort, they found what actually changed is that these epigenetic tags called methylation, they were placed on the promoter region of a particular gene, which had never been described before. So they named the gene acquired cold tolerance, ACT. So what we mean by epigenetic tags is in the letters of DNA, the cytosine letters, you can actually modify the cytosine nucleobase to add a methyl group here. So it's methylated, and it's an epigenetic tag placed on top of the letters of the DNA, which modify how the DNA is read or used. And it's a little bit complicated. I don't want to get into details and make everybody bored here, so I'm going to run through it to get to a simple version of it.
By epigenetic, you mean that something has come beyond the normal DNA that the organism was born with, and this change now has affected their DNA, and they'll pass this trait on to offspring. Is that what epigenetics means?
Well, I think the term epigenetics generically means information that is outside of the simple DNA code.
Yes.
So information in addition to DNA code, and information is replete throughout the whole cell. It's prolific all around the cell. And this is one particular type of epigenetic information where you methylate.
But now it is modifying the DNA.
Yes.
So it's not information that's doing something else, which happens all the time. It's new information is coming in to modify forever, in perpetuity, this DNA, the DNA structure, which would take another modification to ever get it off.
Well, we are finding that these methylation tags can be inherited, so they do go down to the next generation. But they're still learning how long they persist. They don't persist as long as the code itself.
Because we have enzymes that could come and detect them and say, this is a bit out of sequence. Let me pull this out and put a new base in here.
So the gene that they found that is affected here, this acquired cold tolerance gene, a little bit of detail there, it produces an extracellular glycoprotein, which has a lot of effects on the plant. It changes plant growth and development and environmental interaction. So it's really complicated, and they didn't tease all that apart. But what they did do is they found that this methylation process, adding those methyl tags, it is something that represses the use of that ACT gene in warmer climates. So in warmer climates, you have methylation tags that keep this gene from being used. Okay? And they confirmed this by very detailed experiments to take it off, put it on, all that. And they found that the methylation tag is stable and inherited over at least 5 generations. So just to give you a picture here, you have cold weather that decreases the activity of the enzyme that methylates. And therefore you have reduced methylation in this promoter region. And therefore you have increased production of that ACT1 gene. And that produces enhanced cold tolerance. So let me give you an analogy that I think is kind of fun. You have a young child, maybe 5-year-old child, who's headed off to school in the winter. And, they have their coat on, they go to school. Then they go out to recess, and it's really cold outside, and their hands are cold, their hands are starting to hurt. And they just reach in their pockets, and there's a pair of gloves, right? And so the solution to their problem is already there in their pockets. And we all know that those gloves were put into the coat by an agent, right? An intelligent agent, the child's mother, put those gloves into the pockets, knowing that the child might need those during school today. And that's kind of what's happening here. You've got rice in a warm climate that has the gloves tucked away, not being used. But when you get into a cold climate, the gloves can be pulled out and make cold tolerance possible. So it's a set of tools that have been put on the shelf until needed. And that takes foresight. That takes intelligence.
That's actually a nice analogy.
Thank you. So the in a accompanying article to this paper, the authors had this really interesting quote. They said the biggest challenge was convincing reviewers that there were no genetic changes driving this adaptation. Because the reviewers are saying, show me how random mutations and natural selection, show me how genetic changes caused this. And they kept saying, there are no genetic changes. It's something else. Fascinating. Okay, so we're not seeing innovation happening here. We're seeing something that already was innovated with foresight and intelligence, and it's being used to carry the organism through a difficult new environment. Now, this is a little sidebar here also, but this gets interesting. So rice, as we just saw, has been built to handle cold conditions like that. We have the opposite situation with malaria. Here we see malaria parasites eating red blood cells. The most common form of malaria is Plasmodium falciparum, and this is a single-celled eukaryotic, very more simple kind of life, right? It has about 5% of the genome size of rice. So it's a really simpler organism. And malaria is so prolific because, there are millions of people infected each year and other organisms get infected. But for each infection, you can end up with about 1 trillion individual parasites in your body. So we're talking about a very, very prolific kind of life. And why I'm saying that is because there's so much opportunity here for evolution to occur in the malaria. So much opportunity. And yet, for as long as we've had malaria, as long as malaria has existed, it cannot survive in temperatures lower than 61 degrees Fahrenheit. And here's the reference for that. So unlike rice that has this extra toolkit to handle cold, fortunately for us who live in colder climates, malaria just can't handle the cold. And you would think over time it would have evolved because there'd be such a great fitness benefit if it could come up here in colder climates, but it can't. Hasn't been able to overcome that. So it appears that evolution is too constrained, to accomplish this task for malaria, but rice can do it, but malaria can't. I find that interesting.
Yes, that is interesting. That very simple change cannot occur. Evolution is you're convincing me. It's quite constrained.
And, tomorrow there might be a news article that, malaria can now live in colder climates. I hope not. It's possible, but the experiment so far is giving us a clear answer that it's not happening. Okay, so we're going to switch on to another experiment now. Once again, I need to emphasize that these experiments we're talking about here are high confidence experimental prospective evolution studies. This is the best kind of evidence we got. So this should be prioritized over things like the fossil record and things like homology, as we've described. This is the good stuff. This is good evidence we can trust. So this paper came to my mind because of the title. You see, adaptive evolution of complex innovations. That's exactly what we're looking for through stepwise metabolic niche expansion. So I had to read this one. We have complex innovations being shown here. Their goal was to show that complex new metabolic pathways can evolve by a series of small steps. And they started with a strain of E. coli that was not able to metabolize ethylene glycol or propylene glycol. And those are the main ingredients in antifreeze. So that's why I put the symbol here. So they actually started with a mutant form of E. coli that had about 1,000 times the chance of random mutations. The rate of mutations was 1,000 times normal. So they're kind of using their intelligence to up the probability of seeing something interesting here. And what they found was, in that situation, 1 out of 700 million E. coli developed the ability to metabolize propylene glycol, but not yet ethylene glycol. That's interesting. And then they took those successful versions, those mutants that developed the ability to metabolize propylene glycol, and studied them further and found that they could get 1 out of 300 million of those to also be able to metabolize ethylene glycol. So that sounds like a complex evolutionary innovation, right?
Okay, before you go on, let me just make one small mention here, or else I think we're going to get a lot of comments. You said antifreeze, you showing coolant. It's the same thing. It's
Okay.
they both use either ethylene glycol or propylene glycol because that will lower the freezing point and raise the boiling point. So it's the same material. Okay, go ahead.
Yeah, very good. Thank you. Okay, so it sounds like a complex evolutionary innovation until you read the paper and find out what actually changed. That's what we need to know, direct observation of what actually occurred. And they found that if they actually just forced overexpression of an existing gene in the bacteria, the FUCO gene, this actually allowed the bacteria to metabolize propylene glycol, but not ethylene glycol. And then they found that if they overexpressed an additional gene, ALDA here, then the bacteria could metabolize both ethylene glycol and propylene glycol. But you had to have both of them in order to metabolize ethylene glycol. So this is no longer sounding like a great innovation. It's kind of like Lenski's experiment. We're basically just overexpressing existing genes, so no new information, no evolutionary innovation to produce new information, but we do get a new metabolic pathway. And so this quote coming from the paper itself kind of bugs me because they're not providing a rational conclusion. They're saying, taken together, our study demonstrates that complex metabolic innovations can evolve by adaptive means through a step-by-step expansion. When I read this and you're just saying overexpressing 2 genes gets the job done, I don't see how you can call that complex metabolic innovation that evolved. It's just not very innovative, is it? So once again, we're seeing experimental evolution showing us that things are pretty constrained. Even though people want to claim big things happened, when you dig in, it's really not that exciting. So what we're seeing then is these long-term experiments predominantly showing us that we can turn off genes, we can break genes, we can lose regulation, we can lose information. This goes really quite well with what Michael Behe described in his first rule of adaptive evolution, which is that evolution would prefer to break or blunt any functional coded element whose loss will yield a fitness gain. And it's just a simple probability thing. Evolution is going to run by probability and do what's easiest, what's more likely probabilistically. You can also have adaptation occurring through pre-existing systems like we saw in the rice. It could be a complex multi-component system that demonstrates foresight and actually leads to repeatable rather than random mutations. That we see. And we also see single mutations are rather common. But if you need more than one mutation and it doesn't confer a benefit on its own, and you need 2 mutations, then it gets really, really difficult. It's highly unlikely. So we're seeing that true innovation through random mutations is very, very constrained. That's where we're at. Now, what I'm saying here is going to cause a lot of stress, I think, to the evolution community, and they're going to really want to show us something big and innovative because we have all these experiments that we've been through now that are showing us a very constrained process. What's going to come next, I think, this is kind of my prediction, that there's going to be pressure then to demonstrate innovation through random mutations and natural selection, because something needs to be shown here. And as a result, you're going to see the investigators are going to apply increased human intelligence, increased intervention, increased genetic engineering in order to set up the situation just right to get what they want, and very consistent and strong selective pressures to get what they want. And under those conditions, reality can be lost. At what point are the findings more contrived and more human intelligence based than actually what a natural process would do on its own? And I'm sure, Jim, you could speak to this about how in the origin of life community, this same process has been going on. There's increasing pressure, increasing human intervention to try to show something.
In origin of life, there's not a single experiment with massive, that doesn't have massive human involvement. And on an early Earth, there would have been no humans around to be involved. They set the whole thing up. I mean, technically, all you can do in origin of life is to walk into your laboratory and just say, well, I wonder what's going to happen today, and just leave all the chemicals on the shelf and watch something happen. And there's nothing like that. And everything is set up. And even with all its setting up, they haven't accomplished anything.
I think you're going to start to see that more and more here. And I want to show you an example of that. This is the last experiment I'm going to go through. Thanks everyone for being patient and getting to this point. But in this experiment, you're going to see a lot of this kind of intervention. The paper is called A Real-Time Evolution of New Genes by Innovation, Amplification, and Divergence. So these investigators are stating a hypothesis that new genes can develop through this 3-step process. And the way they explain it is that a gene that's already producing an enzyme that has a function, maybe that enzyme can have a second function, kind of a moonlighting extra job that it can do, maybe not great, but it does it okay, And then over time, that gene will get duplicated, so you get multiple copies, and the duplicated genes can then diverge, some of them being really good at function A, and some of them becoming specialized in function B. So it's a nice hypothesis, but of course they're under pressure then to demonstrate some evidence that this is actually happening. And so they do. So they used a lot of human intelligence and genetic engineering, strong and consistent selective pressure in order to demonstrate a proof of concept. Here's how they did it. They take Salmonella bacteria, which has these existing genes, His-A and Tryp-F. We already looked at Tryp-F. That's one of the enzymes used to make tryptophan. And this other gene, His-A, is used to make histidine. So these are 2 amino acids that the Salmonella can produce. And these 2 genes and their 2 related enzymes are really closely structurally related. They have similar functionality. And it's already known that some minor modifications can begin to switch the function between these 2 because they're kind of like sister enzymes. In fact, there's some bacteria that just have one gene, one enzyme that does both of those functions because they're pretty similar. So they said, oh, this is a good system for us to study. Intelligently, they knew this was a good system to study because there's not much of a bridge to build already between these 2. So in the Salmonella, they went in and genetically engineered it to remove the 2 genes and then replace them with one gene that was genetically engineered to be kind of in between. It had 2 mutations caused by humans so that it could partially perform both of the functions. So it's kind of an enzyme that's moonlighting. It's able to do 2 jobs. And they put that gene in the Salmonella in a place that is known to be likely to be duplicated. They also modified the gene so it would be continuously or constitutively expressed. So the bacteria is going to crank out this enzyme constantly, and it's likely to be duplicated. And they also picked an area where duplication is not going to hurt the organism. Okay, so there's a lot of engineering there. And then they applied strong and selective pressure, consistent selective pressures, in order for this bacteria to produce histidine and tryptophan in order to survive and take over the population. So they set it all up, and they do 1,000 generations of the bacteria in this media, and they found that, in fact, the gene was duplicated and 2 mutations occurred. One of them was actually a loss of one of the mutations that they inserted and then another mutation. And waiting through 3,000 generations, up to 2 more mutations occurred. So if you want a generous summary, you could say that gene duplication and 4 mutations occurred, converting one gene into 2 separate genes, which are specialized in 2 functions. So they have proof of concept then that innovation, amplification, and divergence can occur in the situation that they produced. But the innovation really came from human intelligence. They picked just the right target. They removed some genes and put in just the right middle gene that was multifunctional and made it constitutively expressed. And the amplification, they also kind of rigged that because they put it in a location that was going to be duplicated. And the divergence was helped along by strong and consistent selective pressure. So is this really demonstrating evolutionary innovation? How much of it is human assisted and how much of it is natural? This is the strongest example I'm presenting today of what you could call evolutionary innovation. But as you can see, there's some limitations here. So I don't think they would get a patent. I don't think evolution would get a patent for what has been accomplished here. So 15 studies here we've talked about, and they all paint this picture of constrained evolution. And then I want to go back to this where we were saying that we're predicting if this model is true, if one tree of life produced all the great divergence and variety of life we have today, then natural selection and random mutations and natural selection had to be able to produce millions of innovations. And we're just not seeing that. The high confidence, the best evidence we have is giving us a clear picture that this is just not happening. So one more slide to wrap up is the last one. And this takes us back to where we began. Because 166 years ago, when Charles Darwin wrote his book, The Origin of Species, he issued us a promissory note. And he basically said, we don't have a lot of evidence right now back in 1859. But he's saying, I promise in the future, you're going to find strong evidence that spontaneous variation and natural selection are going to produce these biological innovations. Charles Darwin didn't know about random mutations back in that day. He called them spontaneous variation. But here we are, 166 years later, we're still waiting, and we see investigators scratching their heads. We went through these quotes already, scratching their heads, wondering why we can't demonstrate. Why does this still remain elusive? And we have to appreciate in 166 years how much we've learned, how much science has progressed. Back in 1859, this is what a self-driving vehicle looked like. 1859, all right? And today we have a self-driving vehicle. The innovation, the learning, the advancement of science is off the charts, more so in this 166 years than in all of history before 1859. And yet, this promissory note still remains unfulfilled. We are still trying to explain the origin of novelty, and it's still highly problematic. Thank you, everybody. That's all I have.
Well, thank you, Rob. That was great. And yes, evolution is highly constrained, and evolution is quite incompetent to do the many things that people say evolution can do. And when they go in and they load the system to try to do it, even then it does remarkably poorly. And I think that's what you've put forth. So there's another nail in the coffin. And so I've been picking on origin of life. You've been going at evolution. And I think things are going to start to be revised. At least that's our hope. Thank you, my friend.
God
bless.
Jim. God bless. Thank you.
Thank you for joining me today. If you could give us a like, share, or podcast review, we would appreciate it. If you have any questions, you could send them to ask@jesusandscience.org, and we'll try to answer some of those questions in an upcoming video. And if you do not believe in the physical resurrection of Jesus Christ, and you want to hear about why I believe, send me an email to tour@drjamestour.org, and we'll get together by Zoom, and I'll share with you why I embrace the resurrection of Jesus Christ.